Hello, everybody, and welcome back to BioSC 140 Human Physiology. This is the discussion video for Lab 6. All right, so let's start off by saying, remember, there are two parts to Lab 6. There's the DNA extraction with the strawberry and the electrophoresis. Let's start with the DNA extraction. So you should have already watched the first video and you should already know the procedure for this experiment. I want you to know the procedure for this experiment for the quiz. I want you to know why we did each step for the quiz. And that's what we're going to go over right now. So explanation of materials used. Tissue. If we use mammalian tissue, such as thymus or testes, it would need to have a high nucleus to cytoplasm ratio and not contain large amounts of lipid and carbohydrate. The extraction that we did was a fairly simple, easy extraction. As long it's only possible, such a simple extraction is only possible because we chose the right type of cells, the right type of tissue to use. It requires more complicated steps, and the yield is not as high as it is sensitive to environmental conditions, so it's simpler to extract DNA from plants. It's easier to get DNA from plants than mammalian tissues. Ripe strawberries are an excellent source for extracting DNA because they are easy to pulverize and contain enzymes, where enzymes cause reactions to take place. They lower the activation energy, allowing reactions to proceed faster. Pectinases and cellulases, those are types of enzymes that help break down cell walls. Fruit contains enzymes that in part break down fruit into simpler molecules. And we're using those enzymes that are naturally present in strawberries to help release the contents of the cells that we need. They help break down cell walls. Perfect. While commercial strawberries only have 7 chromosomes, through selective breeding, they have 8 copies of each chromosome. So they have 8 copies of each chromosome. There's plenty of DNA to isolate and observe within a strawberry. So that's why we chose a strawberry. They're easy to obtain. They have a good nucleus to cell ratio and size. They have a lot of DNA present within them. They have pectinases, they have enzymes within them that help break down the structures within them. Enzymes like pectinases and cellulases, which allows the DNA to be released, to be accessed more easily. The extraction buffer. So first thing we did in this lab was we smushed the strawberries. After that, we added the extraction buffer. The extraction buffer contains a soap. Soaps they contain detergents, which are amphipathic molecules. An amphipathic molecule has a significant nonpolar side and a significant polar side or region. Soaps disrupt cell membranes. They break cell membranes apart. So the soap helps dissolve the lipid membranes of the cell, which releases the DNA into the solution. The cellular membrane is a lipid membrane that soap helps dissolve, and so is the nucleus. The nucleus has a lipid membrane that soap helps dissolve. There's also sodium chloride, or a salt, within the extraction buffer. Sodium chloride helps keep the DNA molecules together, as the salt contains sodium ions, which stabilizes the negative charges from the phosphate groups in the DNA. Salt also removes the proteins that are bound to DNA, and it helps to keep the proteins dissolved in the aqueous layer so they don't precipitate in the alcohol along with the DNA. After we added the extraction buffer and mixed it up with the strawberries, we strained it through a cheesecloth to get the solids out. And then we slowly added cold ethanol. DNA is soluble in water. DNA is not very soluble in ethanol. Since DNA is not very soluble in alcohol, it precipitates out of the solution so that we can easily visualize it on the spool of DNA. So precipitate. That means comes out of solution. It goes from being a dissolved molecule to a not dissolved molecule, a solid. You can see up here that it is no longer dissolved. You can see it on the spool, on the piece of wood right here. It is no longer dissolved in the solution. It has come out and become solid. So DNA, when it's in this lower portion, this aqueous liquid portion, the DNA is dissolved. Now, think about what's going on in chemistry. The DNA is bouncing around, it's moving around. Any DNA that comes to this border with the ethanol and moves into the ethanol, once it moves into the ethanol, it's going to come out of solution. It's going to become not dissolved. It's going to precipitate out and become that gooey solid that we see in the top ethanol layer. So that's the reason for the ethanol. The reason for the ethanol is because DNA precipitates and it becomes not dissolved. So we're able to isolate it and hold on to it. Did you get what appears to be DNA? Well, I mean, you don't see like that perfect double helix image. We get something that's, students describe it usually as like mucus or like gooey spider silk. Perfect. Why do we use the extraction buffer instead of distilled water for this experiment? Well, the extraction buffer contained two things that helped us with this experiment. It contained a detergent, contained soap, it contained an amphipathic molecule which helped dissolve the lipid membranes, the cell membrane and the nuclear membrane, which released the DNA. And it also contains salts, and the salts helped stabilize the DNA molecule, and it also helped remove any proteins bound to the DNA molecule. What are the components? Just went over that, perfect. What was the role of ethanol in the DNA extraction experiment? The role of ethanol in the DNA extraction experiment is it took DNA from a dissolved molecule to a solid, to a not dissolved molecule, to a precipitate. It brought the DNA out of solution and made it a solid substance. What are the advantages of using strawberry DNA over mammalian DNA? Well, first off, it's easier. I don't want to donate my DNA to this, to this experiment. It's much easier just to go buy some strawberries. Strawberries also have more favorable biomolecule components, makeup. It doesn't have as much fat as a human cell. And it's just, there's less steps needed to process it because there's fewer of those extra biomolecules. Strawberries have a very favorable DNA to cytoplasm ratio. They have a lot of DNA. Commercial strawberries have 8 copies of each chromosome. That's a lot of DNA. So there's a lot of DNA to collect. Strawberries also contain enzymes, and they contain enzymes that help break down macromolecules within the strawberry itself. So it further makes it easier to process and extract the strawberry DNA. Next up, we're going to talk about the gel electrophoresis. So let's talk about this setup. Let's review the setup again. So we have this gel, electrophoresis gel. It's a lot like Jell-O. We're going to have a few holes cut in it called wells, and we're going to put samples of DNA into each one of those wells. We're then going to take our gel and we're going to put it inside the electrophoresis chamber. The electrophoresis chamber is going to be filled with a salty water. We need salty water because electricity does not travel well through pure water. It travels much better through salty water. And then once we have our gel and our samples in the salty water, we're going to run electricity through it. We're going to put the wells that contain our DNA samples on the negative side of the chamber because DNA is negative and it's going to move towards the positive side. So our DNA samples are going to get pushed and pulled through the gel towards the positive side of the gel. Now, before we get into exactly what our DNA samples are, let's talk about a few things about DNA in general, about DNA sequences. Let me tell you what we're going. Let's just start off here. So let's talk a little bit about DNA. So harmless mutations are useful genetic identifiers. So all of us, provided there's not identical twins in this class, have our own unique DNA sequences. We all have our own unique DNA. All of us have variation. Within this class, there's variation amongst our DNA. And there's different types of variation. This top one right here, copy number variation, CNV, all of us have sections of our DNA code that have many, it's like the same sequence repeated over and over and over and over again. Now these sequences, these sections of DNA usually don't code for any enzymes or any molecules. It's just like filler space within our DNA. So having different numbers of these CNVs doesn't really change the function of our DNA. But there's variation in CNVs amongst all of us. So you can see here on the right examples. So we have copy number variation. We have allele 1, where there's 1, 2, 3, 4, 5 copies of this variation within this strand. And then down here on the bottom, there's 2 copies. There's 2 repeats of this sequence. Now, the sequence may be AT. The sequence may be GT. The sequence may be any number of sequences, but it's repeat sequences, repeat segments. This top strand has 5 of these repeat segments, so AT, 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 AT. And this bottom one has 2, so AT, AT. That's one source of variation in DNA strands between people. Down here, we have something called a single nucleotide polymorphism, an SNP, single nucleotide polymorphism. It's an individual nucleotide difference. So it's one nucleotide in your DNA code that's been changed, a single nucleotide polymorphism. So poly is like many, morph is like form, so many forms, a single change. Comparing this top strand of DNA with this bottom strand of DNA, you can see they both have G in the first position, T in the second position, A in the third position, and then there's a single nucleotide polymorphism between the 2 alleles, between the 2 strands. We have a C in the top one and a G in the bottom. And then they both have G, they both have T, they both have A, and they both have C. This is another source of variation amongst our DNA. Restriction enzymes. I'm going to tie this all together, but I want you to get what a restriction enzyme is. A restriction enzyme is an enzyme that will cut a DNA sequence, a DNA molecule, at a very specific sequence. It will cut a DNA molecule at a very specific sequence. It's going to move across your DNA molecule until it finds a specific sequence. In this example, that sequence is GAATTC on the 5 to 3 side of the double helix. when the restriction enzyme sees that sequence, it's going to cut the DNA. The spot where it cuts the DNA is called a restriction site. It's called a restriction site. And there's many different restriction enzymes, and each one's going to cut the DNA at a unique restriction site. There's many different restriction sites. Every different restriction enzyme will have a different restriction site. And the way that it cuts the DNA can vary also. So you can see this one, it cuts in between the G and the A, goes along the strand then to the CG and cuts right there at the other side. So you get these little sticky ends hanging off. Now let's combine the last two things that we really talked about. We talked about genomic variation and we talked about restriction enzymes. Let's see how that plays out together. So let's take a hypothetical situation. We take a hypothetical situation down here in the bottom right corner of this screen. We have strand of DNA big A and strand of DNA little a. And we expose both of these strands to a restriction enzyme. So both these strands are exposed to the same restriction enzyme. That restriction enzyme is going to identify this spot right here as a restriction site to cut the DNA. Remember, both these strands, big A and little a, the DNA sequence is the exact same. So big A and little a, this DNA sequence is the exact same except for this point mutation, single nucleotide polymorphism right here. That's the only difference between these two strands. So the restriction enzyme is going to identify this spot as a spot to cut the DNA. So both DNAs get cut right here. It's going to recognize this spot as a restriction site. Both DNAs get cut right here. It's then going to continue along the DNA molecule to this point right here. On big A strand of DNA, it recognizes this spot right here as a spot to cut the DNA. Because little a DNA has a different nucleotide at that point, it does not cut the bottom strand. The restriction site was altered, and it was altered in such a way that it makes that restriction site not a restriction site. So there's no cutting of the DNA at this point. Now I want you to look at the pieces of each of these DNA. So the fragments of this DNA after it's been exposed to this restriction enzyme. You're going to have this little piece here on both of them. That's going to be the same between the two. You're going to have this piece in between the two restriction sites on both of them. That's the same size also. If you look at this part, the right half of each of these DNA strands, the bottom DNA strand, little a, is going to have one large uninterrupted piece. One large uninterrupted piece. Big A strand is going to have a medium-sized piece and a smaller piece. A medium-sized piece and a smaller piece. Restriction enzymes, when you expose the restriction enzyme to each one of these strands of DNA, it will cut those DNAs at different spots because of the single nucleotide polymorphisms, resulting in different sized fragments. 3 fragments for little a, 4 fragments for big A, different size for the fragments. So now let's take a hypothetical situation where we had a few repeat segments. Let's take this repeat segment and put it right here. Take this repeat segment, little a, where there's 2 repeats, and put it in between these 2 restriction sites. Now take big A, 5 repeat segments, and put it in between these 2 restriction enzymes. The presence of varying numbers of repeat sequences of copy number variations in this segment of DNA would result in different sized fragments of these two pieces of DNA, even though the restriction enzymes still cut at the same code. Because there were 5 repeat segments in big A between those restriction sites and only 2 repeat segments in between the restriction sites in little a DNA, you would end up with different size fragments. Well, with all that in mind, what we're doing with our DNA samples in the gel is we're taking DNA samples, we're exposing those DNA samples to restriction enzymes, which cut the DNA into fragments, smaller DNA fragments. We are then going to put those DNA samples in these wells, and we're going to have the electrophoresis pull the DNA through the gel. Pull the DNA through the gel. And what that's going to do is it's going to separate the DNA by fragment size. So this gel is like a web. It's like a at a molecular level, it looks like a big clumpy spider web. Smaller pieces are going to get pulled faster through the gel. Larger pieces are going to get pulled slower through the gel. You can look at the bottom fragment, the bottom example here. You can see how there's these bands that form. All that DNA started out in the wells, and as it gets pulled through the gel, smaller pieces are going to move faster, larger pieces are going to move slower, and it's going to separate our samples by fragment lengths. Now, all of us have single nucleotide polymorphisms. So all of us if we expose each one of our DNAs to a restriction enzyme, there should be variations in where our DNA gets cut. Because we all have copy number variations of repeat segments within our DNA, even segments of our DNA where the restriction enzyme might cut in corresponding spots, because of different numbers of repeat segments of copy number variations, we're going to have different size fragments. So it makes sense that each one of us, if we all exposed our DNA to the same restriction enzyme, the size of the fragments of DNA that resulted from it would be different. So let's say we had an unknown DNA sample from a crime scene, and we have 2 suspects. We don't know who the 2 suspects, we don't know which one of the suspects was, actually had the DNA at the crime scene. Well, we could expose crime scene DNA with DNA from our 2 suspects. We could expose their DNA to a restriction enzyme, run it through a gel like this, and if the DNA at the crime scene matched one of the suspects, they should have the same fragment size of DNA. And the other person should have a different banding pattern. So that's really where we're going with this. So, all right, restriction fragment length polymorphism. It's underlined and bolded. I've actually asked on quizzes before, what does RFLP stand for? So just a heads up, just one point. What does RFLP stand for? I probably won't ask that now, but be ready. 1 second. All right, I'm ready. So polymorphism. So poly meaning many, morphism means form. So variation in DNA sequence. Affects restriction sites and affects the fragments produced. So restriction fragment length polymorphism. This is restriction fragment length polymorphism. It's exposing DNA to restriction enzymes and getting different sized DNA fragments out of it. So let's look at another example. So right here we have the normal beta globin allele. Beta globulin allele. Each one of these kind of down over down lines, like each one of those is a restriction site. We expose this allele, this piece of DNA, to a restriction enzyme. We get a cut right here. 175 base pairs later, we get a cut right here. 210 base pairs later, we get a cut here. And then we get a large fragment on the end of the DNA strand. Sickle cell anemia is something that you'll come across in the healthcare world, in our society. It's present. And sickle cell anemia is caused by a single nucleotide polymorphism. It's caused by a single change in this allele, a single nucleotide change. And because of that single nucleotide change, the restriction enzyme does not recognize this restriction site right here to cut. So there is no cut at that corresponding spot when exposed to a restriction enzyme. So we get a cut here, and then 376 pairs later, we get another cut in the DNA. Now imagine we had both of these samples right here, and we put the top one in a well and moved it through the gel, and the bottom one in a well and moved it through the gel. We would have two bands corresponding to we'd have one, two, well, just right here, this section right here, we'd have two fragments that would move pretty far across the gel. And right here, we'd have one larger fragment that wouldn't move quite as far. We would have a different banding pattern on the gel itself because of that single nucleotide polymorphism, because that single nucleotide polymorphism made this restriction site not a restriction site. So right here, we can see how it looks. We have our two bands right here. All these bands, they all started out right here in these wells. We have our normal allele, and we have our large fragment there, and then we have our 201 base pair and 175 base pair pretty far down the gel in two distinct bands. And then the sickle cell, we just have our 376 base pair long fragment right here. And right here is a sample of an actual like gel readout. So this is a little dated. This isn't how they do it nowadays, but not too long ago, you know, if there was DNA at a crime scene and they need to compare that DNA at a crime scene to two suspects, this was commonly done. This was done in courts of law. So right here, we have a gel electrophoresis. Let's go through each one of these wells, each one of these rows. So we have the victim. Right here, we have victim DNA. Right here, we have suspect DNA. Suspect 1 DNA. Right here, we have suspect 2 DNA. Right here, we have female cells that we found at the crime scene. Female cells found at the crime scene. Right here, we have semen that's found at the crime scene. This well is called the size reference. So a lot of times when you run these gels, you'll put in what's called a ladder, and the ladder has like different size DNA fragments that are known sizes. So if you know the largest size is 1,000 base pairs, the next one's 900, the next one's 800, you can kind of guesstimate how large the fragments are to the sides of it. And then right here we have boyfriend DNA, victim's boyfriend DNA right here. So we exposed all of these, except for the size reference, but all the samples of DNA to a restriction enzyme. And then we ran it on a gel electrophoresis. Victim DNA right here matches the female crime scene DNA. So we can confirm that the victim's DNA is the DNA that we found at the crime scene. Right here is the semen from the crime scene. We can confirm that it's not from her boyfriend. We can look at suspect 2, different banding pattern. We can confirm that it was not suspect 2's DNA found at the crime scene. But what about suspect 1? Suspect 1, his DNA pattern is the exact same as the DNA pattern for the semen. So this would be pretty good evidence that the semen found at the crime scene came from suspect number 1. This banding pattern is sometimes called a DNA fingerprint. Sometimes called a DNA fingerprint. And it can be used in crime scene identification. And it's gotten people convicted, and it's also exonerated people. Right here, Mr. Washington was released from jail in 2001 because gel electrophoresis proved it was not him. All right. If you have any, let's go back up. We have some questions that we should go over. So, define RFLP and give their significance. Restriction fragment length polymorphism. So, restriction fragment length polymorphism, the significance is when you produce them, when you expose DNA to restriction enzymes and then run it on a gel electrophoresis, you get a unique banding pattern. You get a unique pattern for each person. Every person should have a different one. And you're able to, identify a sample of DNA or compare it to people and figure out if that sample matches them. Over here on the right, this is actually what it looks like when we do our analysis in lab. In fact, this photo's from the lab supplement. This is a photo directly from our lab. List the steps involved in DNA fingerprinting, from extraction through to autoradiography. I'm not going to ask you guys to list all the steps. Just know what we've talked about here in this discussion. Taking your DNA sample, exposing it to the restriction enzymes, running it on a gel, comparing different samples. Explain the significance of repeat DNA sequences. So what's the significance of repeat DNA sequences? Well, they're going to cause for they're going to produce different fragment lengths. They're going to be the reason for different fragment lengths when exposing different people's DNA to restriction fragment length or to restriction enzymes. Right here. If there were if there are 5 tandem repeats, 5 repeats right here, and 2 repeats right here, these fragments would be different lengths. Who are the only individuals possessing the same DNA fingerprints? It'd be identical twins. They have the same DNA sequence. And what type of human cells can be utilized? The types of human cells that can be utilized for this technique, ultimately, different types of cells and different types of tissues are going to be easier than others, but you need to have a human cell that has DNA. Now, we talked some about crime scenes. What's a common thing that people, see at crime scenes maybe? Blood. Red blood cells, mature red blood cells do not have a nucleus. They do not have DNA. We'll learn all about that when we get into the cardiovascular section of this class for test 3. But mature red blood cells do not have a nucleus. They do not have DNA. So you cannot use red blood cells for this. You have to use some sort of cell that has DNA. So red blood cells, no DNA. Any cell that really has DNA, different tissues are going to require different techniques, but that'll get you there. If you have any questions, please email me. Have a great day.